Yep.
It's about 1:30 Pacific Standard Time. So, hello everyone. This is the Diagnosis webinar titled Biologist Introduction to Label-Free Proteomics. Um, and today, the speakers are myself. My name is Christopher Balaktis. I'm currently the West Coast Business Development Manager. A little bit about my past. A biologist and also a immunoassay development scientist. I used to work for Bayer as well as Novartis. I'm also joined by Dr. Jeffrey Silva, who is currently our Director of U.S. Business Development. Um, Lots of proteomics experience. In the past was the Director of Proteomic Services at Cell Signaling Technologies, as well as a proteomics senior scientist at the Waters Corporation. So today's agenda, the webinar is about uh, 1 hour, and the first 30 minutes will be introduction to diagnosis, and then the last 30 minutes will be answering questions that you might have based on our webinar. And if you have any questions during my talk, if you can please add them to the chat box in the GoToMeeting section, and myself as well as Jeff will be happy to answer your questions. So let's begin. Um, Diagnosis was founded in 2008. It's a spinoff from Professor Rudy Abersol's lab, who is a leader in next-gen proteomics, also founded by our CEO and founder, Dr. Oliver Reiner, who's also from the Abersol lab. And at Diagnosis, our mission is to make next-gen proteomics widely available through contract research services, as well as offering innovative reagents and bioinformatics software. Currently, Biognosis is located in Zurich, Switzerland, and because of the growing demand of our technology and services, we've also recently built um, a US BD team in Boston, Massachusetts. So at Biognosis, our core strengths are the unique technology that we provide, being a co-inventor of next-gen proteomics. This method is a label-free method, which limits the sample loss and quantitative variability. Another strength is our flexibility, speed, experience, and using high-end instrumentation for our services. So for proteomics or for protein analysis, there's definitely a gap between being able to identify a number of proteins as well as being able to quantify or reach a certain level of sensitivity. This is primarily done using antibodies, such as an ELISA assay. And then with classical proteomics or shotgun proteomics, what we see is there's basically a gap between being able to detect a large number of proteins and having an effect on the sensitivity of that protein detection. Here comes what we've started off doing, which was targeted proteomics, primarily HRM assays, where you're able to increase the sensitivity as well as increase the number of proteins you can detect. And the unique technology that Biognosis offers is around what we call next-gen proteomics, which is HRM. HRM stands for hyperreaction monitoring, where we're able to close the gap between scale and accuracy, seeing technical CVs at around 10%. That enables us to look at thousands of proteins with high precision and throughput. So a little background information and principles of the technology that we use. It's using the mass spec instrument, where where proteins are large molecules, and in order to get them into the mass spec, we have to cut them into peptides using proteases such as trypsin. The peptides then act as small molecules, which they're very stable. They're about 5 to 25 amino acid. Um, and these peptides become proxies for protein quantification. The peptide sample is then injected into an LC mass spec system. The LC, as you can see, separates the peptides and then ionizes them as they enter into the mass spec. It reaches the first level of the mass spec to get to generate a intact peptide spectrum. This provides information about the abundance, quantification, and in order to get further characterization and identification of that peptide, we fragment it into MS level. So with the differences between classical shotgun proteomics compared to next-gen proteomics is we can see here in classical shotgun proteomics, the mass spec first identifies usually the most intense peaks. Those most intense peaks are then generate the spectra, and then those spectra then are used to identify the peptides. The advantage of it is that you get a one-to-one ratio between the spectra and the peptide you identify. However, the disadvantage is that you start to is that you don't identify the low abundant peptides that are coming on the mass spec. Here is where the advantage of using Biologist is HRM technology, where we look at a comprehensive identification of all detectable peptide ions. This is done in parallel, doing a parallel fragmentation process, where we broaden the precursor windows, and what we do is we look at all of the fragmentation peaks, which generates a very convoluted spectrum. However, using our bioinformatics software, we're able to deconvolute that spectrum to do identifications of the peptides. So in a real world example, more of an analytical comparison between shotgun and HRM technology. Here on the y-axis, you look at protein abundance, and then using the same HEC293 cell lysates, we did 24 repeat injections. And in shotgun proteomics, you see this leads to incomplete and irreproducible data. This is sort of represented in the white blank spaces, whereas using HRM proteomics, you see a more complete data set, which is more reproducible, which enables a biologist to do more interesting types of experiments, such as time course studies, dose-dependent studies. For our services, we can handle various types of sample matrices, such as cell culture, tissue, urine, CSF samples. And here's a graph basically showing the typical number of proteins we can identify, the amount of samples that we require, and whether or not we're able to do enrichment steps to look at PTMs. We also have other service offerings as far as offering longer gradient lengths to get deeper coverage, to see more proteins. We have in-house depletion methods and can also do depletion methods that the customer needs. And finally, we also offer fractionation. So for our services, what we try to do is to get a better understanding of the research questions you would like to ask in order to identify which proteomic strategy is best for you. We then have a scientific project manager which prepares a project proposal, and then once your samples are ready, the samples are shipped to our diagnosis site, where we provide a shipping protocol. Samples are typically frozen, and because the CVs are very tight, we typically prefer a biological replicates. Once we get your samples, we do the protein extraction, digestion, and cleanup using our own sample preparation kits. During the data acquisition, we use calibrators such as our own IRT kit, which is critical for using our own Spectronaut software for the data analysis. So what you get once a project is completed, of course, is a project description and summary report. You'll consult with our scientists as well as the scientific project manager, where we will, of course, give you a list of all detectable proteins and peptides, along with the proteins that regulate up and down at least a 50-fold fold change. This can be represented also as a heat map overview or volcano plots. And we can also do customized analysis reports, such as pathway analysis. So to go into the different types of case studies that we've done, one that I'd like to focus on is a discovery proteomics project that we've done that was also published, and that's the toxicology of 3D microtissues, doing a drug treatment study with acetaminophen. So with this case study, we did a drug treatment with acetaminophen on 3D human microtissues with our collaborator at Inspiro. These microtissues have liver-like morphology, and what we did is we started with acetaminophen with a 72-hour exposure at different concentrations, biologically relevant concentrations, which you can see are the concentrations at subtoxic as well as toxic concentrations. Here we have the cell viability assay, and basically the concentrations in the dark red circles are the concentrations we used for our study. And also an interesting point for this study is that we only had to use 12,000 cells, which was about 20 micrograms of total protein. So from the proteomic view, this is sort of a needle in the haystack, where what we're looking at is fold change on the y-axis and different concentrations on the x-axis, and you can see a lot of proteins regulating up and down. After doing t-test analysis, we can look at lower concentrations of acetaminophen and seeing that roughly 100 proteins are regulating, and as you increase your concentration of acetaminophen, you see more proteins changing at higher concentrations. Typically with these types of studies, we look at ADME-tox type of functional assays. However, we're looking at it at the protein level with at phase 1, 2, and 3. For instance, at phase 1, looking at P450 induction, looking at the CYP proteins. This is one way that can represent the data that you get from a proteomic analysis using HRM. Looking more closely from an analytical point of view, in this particular study, we did 3 replicates, and this is the results from a CYP1A2 study. We have the 3 replicates here at different concentrations of acetaminophen, and if you're used to looking at MRM data, you can see that HRM data looks very similar as far as being able to generate the concentrations based on peak intensities. Here is at phase 2, looking at this UGCA1A1 protein. Again, our typical replicates are very tight, so in most cases we only need biological replicates to do a service project. An interesting thing, because of the HRM technology, is being able to look at all of the proteins that come off the column. We're able to look at specific proteins you want to look for, and also proteins that you might not have thought of looked for before you started the experiment. In this case, one of the questions that our collaborators wanted to ask was, they knew that at high concentrations of acetaminophen, adducts form on proteins, on cysteines. However, they weren't able to identify which proteins the adducts formed at. Going back into the data we were able to identify at least 6 proteins where the adducts were forming. And in this case, we're showing the data for PARC7, where NAPQI adduct formed at this concentration, and the non-modified PARC7 protein, as you can see, starts to decrease. So you can start to develop a stoichiometric relationship between adduct and non-modified protein. And at the same time, look at control proteins and see that they're roughly the same. So that concludes the presentation part of my talk, and now I would like to open it up to the questions you might have.
I think before we start with the questions, I'll just ask a couple polls. Let's see if I can actually, I'm just going to put this up to see if of the people that are listening, what types of samples they typically use in their studies. We'll collect the data for maybe a minute just to get a sense of the people that are listening, the types of samples that people are typically working with. About 30 seconds, we'll shut this off. And then if you have questions, you can provide questions here in the question section of the control panel. 10 more seconds, we'll have the poll. Looks like a majority of our listeners are working with cell lines. Then we have tissues and xenografts. I'll close the poll. Thank you for responding. So we have a question here. Do you have other examples looking comprehensively at the off-targets of covalent drugs from cell culture? For example, acrylamide, warhead-bearing covalent kinase inhibitors. I guess what I'll say is that this acetaminophen study was an example just to show that the beauty of the data-independent acquisition, or HRM, is it archives all of the information from your sample. And so if you have queries about a particular adduct, it's just a matter of searching for that particular peptide adduct mass. As well, it also gives you the ability, because you're collecting this DIA data with the fragmentation data, you can actually not only query the precursor, the intact adduct mass, but the fragment ions that are associated with that to confirm the sequence in the adduct product. Do we have other examples? I'm sure we can look into this and get back to you, Michael. I would say that perhaps follow up with an email and we can look into providing you with some other examples. I hope that was at least helped you with that particular question. We've got another question here from William. Is there a preferred MS platform, QTOF or Orbitraps? The QTOF version of this DIA method is termed SWAF. I would say that there is no preferred method. I can say that from Biognosis's perspective, we've enabled the labs with Orbitrap instruments because it is based on our analysis, our preference. We have decided to work with the Orbitrap instruments. However, QTOF SciAx instrument is also a platform that can be used for this as well. So thank you, William, for that question. There are some other questions as I'm waiting for other people to reply. I'll come up with a couple other questions until I hear them from participants. Here's a question that has come up often. Do you have a preferred license buffer that I should use for sample prep? In terms of doing services with us, we actually prefer that the client send us either samples that are either flash frozen, so in regards to tissue, flash frozen tissue, or just simply PBS washed cell pellets, and then we would carry out all of the sample prep, including lysis, all the way through the data analysis, data acquisition, and data package generation. So to minimize the sample handling, we would prefer to do all of the sample prep. However, as Chris mentioned in his talk, Biognost does have sample prep kits that you can use if you are doing this yourself, or you would like to prepare the samples yourself. Let's see, another question. Can we use this type of method with enrichment samples? If, for example, you're doing studying complexes where you can do an IP for a particular complex and look at different conditions, treatment conditions, or cell lines, knockouts for the presence of complex components. The answer is yes, because HRM allows you to interrogate the DIA files across all of the different replicates. Chris, would you bring up that DIA shotgun slide? It's a great method to use for these types of applications because you're able to extract intensity information from all of the samples. If you happen to miss an MS identification in a particular sample through shotgun, because of the HRM, you would pull out the intensity measurements across all of the different samples. So it's a really nice way to prepare the samples, collect the data, so that you can interrogate the data with a full data matrix. Looks like the slide isn't coming up yet. Let me know, Chris, when you have that up.
Okay. Is this the one right here?
No, I'm thinking the one with the shotgun versus HRM. The duty cycle slide. I guess we'll just move on to the next question. I have one here from Martin. Do you need to build your own spectral libraries for your instrument, for your own instrument column, etc. system, if you want to run the program yourself? If so, does your program build libraries? So typically, when we do a study for a client, what we'll do is if a client, for example, has 3 different sample conditions, and this will be dependent upon the study and the goals and objectives of the study, but we would build a spectral library from each sample condition. That spectral library, each of the samples are prepared in a way where we fractionate. We pool, if there are biological replicates, we'll pool those biological replicates and then run a high pH, low pH 2D fractionation of that material to build a comprehensive spectral library of each of the sample conditions, and then from that compile put together a complete spectral library that can be used now to interrogate the DIA acquisition files for each of those identified peptides. And in this slide that's up here, this is what gives you those complete data sets where the data matrix itself is filled with intensity values because you're interrogating that that DIA file, where with shotgun, if you don't switch on that particular peptide in that particular sample, you're not able to get an intensity measurement. This is especially true for TMT type methods where, for example, if you've got for TNT10, for example, if you've got a study where you have 20 or 30 samples, it forces you to do the experiment in 3 separate TNT experiments. So those are separately fractionated, and if you happen to miss the identification of a particular peptide in one of those 3 samples, what will happen is you'll end up with holes in your data that for that particular sample set that could be 10 samples wide, right? Because you've been forced to have that second reaction, the third reaction with 10 samples in it. HRM, the DIA method, doesn't have a limitation in terms of the number of samples that you can put in. We've done studies with 100, 200 samples. We don't have to limit the size of our studies based on the labeling strategy for the in TNT type methods. So I hope that addresses your question. Thank you, Martin. Let's see. There's some other questions that we'll wait for other questions to come in. Here's a question that has come up before. What's the typical sensitivity or dynamic range for an HRM assay? Typically, with regard to dynamic range, we can easily get 3.5 to 4 orders of magnitude dynamic range. And this is obviously dependent on the peptides, but the dynamic range for this particular assay is quite high in terms of sensitivity. Can easily detect low femtomal quantities of peptide on column. Let's see. Here's another question that has come up. Type of quantitative variability should I expect from analytical replicates? Typically, the CVs for analytical replicates in these types of methods are easily under 10%. Biological CVs will obviously vary depending on the model system. Cell lines are tighter because you're able to control those growth conditions in the tissues. So cell lines are typically, in a well-controlled system, can be 20 to 30%. Tissues are, in terms of biological variability, can vary depending on the model system as well. But something that's normal is typically 20 to 40% biological variability in nicely controlled systems. Let's see. Can you provide absolute quantitation for protein targets with this platform? Is another question that's come up. We can conduct these studies with a heavy isotope labeled peptide. So if you happen to have a set of targets within your study, let's just say you would like to do a discovery experiment where you want to identify and quantify as many things as possible. Maybe it's you're looking at a series of drugs and you want to try to differentiate the mechanism of action across the different samples, drugs, I mean. But you have a core set of proteins that you'd like to quantify with absolute quantitation. So what you can do is you can include a heavy label peptide for each of those targets. Maybe that's a subset of maybe 10 or 20 proteins. We can include peptides for each one of those proteins in the assay, and and determine the absolute quantitation, whether we collect it by HRM or we can configure that in an MRM as well with a triple quad and give you more sensitivity for that particular assay for those protein targets. Here's another question that's come up often in terms of normalizing the data. How is it normalized? This is a standard methods that are typically used for global proteomics is similar to that used in gene chip transcript arrays. Basically, it's a median normalization technique where each of the we do a distribution of all the binary comparisons. The assumption is that most of the things don't change, and so what you'll get is a distribution that should center at log 0, log ratio equal to 0. And if it doesn't center at 0, what we do is we apply an offset correction to readjust that distribution so that it does center at 0. And that's a typical normalization strategy, a median offset correction technique that's used in proteomics as well. So I think that actually we are close to our time. I'd like to, Chris, if you want to wrap things up.
Mm-hmm. Yep, thank you everyone for attending our webinar, and just to let you know that our next webinar, which is going to be more focused on targeted proteomics, is going to be September 27th. We can send the registration link to those who attended the webinar. Also, this presentation will be made into a PDF and sent to the attendees as well. Okay.
Thank you all for your time.